Great. So it's great to see everyone here. And I'm going to briefly talk about, not that briefly, quite briefly talk about where Mendelian randomization came from and just finish with a very few speculations on where it might be going. So Hugh mentioned, we call this the 4th International Mendelian Randomization Conference. This one was the first in 2005, and it was at the Mendel Museum. Sadly, Mendel was only represented by a statue, but he was there for this meeting. And also, sadly, quite a lot of people at this meeting are no longer with us, but there's quite a few old friends who are still working in the area. And there's probably one person that people from the IU will recognize. This is this young man was going around, and Shah Ibrahim, who helped organize the meeting with Paul Brennan, did say that Nick could go as a PhD student, but he did hand out the canapes, so you would have seen Nick Timpson, now the director of ALSPAC, was handing out the canapes at the meeting. And we've had biannual meetings over the last period, and this is the third of the meetings since we started the biannual meetings. So where did Mendelian randomization come from? Well, it obviously came from Gregor Mendel, but its foundation comes from Mendel's laws that you are all taught about in school. And like everything which has a name attached to it, of course, Mendel's laws were not derived or put forward as those laws by Mendel. Mendel's laws were actually put together as Mendel's first and second law by the genius of Robert Heath Locke, who sadly died much too young, a founding British geneticist and follower of Bateson. And in 1906, Locke extracted out of Mendel's extraordinary paper, which I recommend very highly to everyone. There's probably about 7 things which could be called almost law-like statements in it, but there's no laws. But the 2 laws that Locke extracted and said was the essence of it, which has now become what we think of as Mendel's laws, and people quote these and say that they're in the paper. Extracted from that 2 laws, both of which Mendelian randomization depends on an adapted form. The first law is of segregation, which is that the germ cells separate, and then you've got to think, for Mendelian randomization to be true, that those germ cells will go through to conception, they will go through to live delivery, and actually will go through to entering your study independent of the environment. Now, there's been lots of literature on possible segregation distortion, as it's called. For example, suggesting that women who have low folate levels are more likely to select out fetuses carrying particular genetic variants. So there's no reason why it has to be true. It should be something which requires to be demonstrated. And huge parent-offspring cohorts now allow people to start looking at this assumption. So it depends on segregation. It depends on the one that people usually write down. So many MR papers say, Mendelian randomization because of the second law, because of independent assortment, i.e. one gene sorts independently of another gene. That doesn't say anything about environment. So, but it is important in keeping, obviously, your genetic variants, except that those that are in linkage disequilibrium don't confound each other. So, but those are the principles on which Mendelian randomization is based. And both actually can't just be asserted. One does need to try and demonstrate them. Okay, the second progenitor, although not one that I think people were thinking about when Mendelian randomization was initially being discussed, but certainly in retrospect, is Sewall-Wright. Sewall-Wright introduced path analysis. And path analysis is a sort of form of a causal diagram that has been adapted into what you now know as directed acyclic graphs, as Judea Pearl and others discussed in terms of the origins of causal graphs. And this And these are then used for trying to make causal inference from non-experimental data. Now, Wright was absolutely explicit about one thing, is that these were only meaningful because you had causal anchors. And the anchors in virtually every one of Wright's very many examples of use of path diagrams was Mendelian inheritance. So here is Wright responding to some criticisms that you can't get cause out of observational data. He says that this rests on the validity of the premises, i.e. on the evidence for Mendelian heredity. The paper began with a quotation from Easton Jones on the universality of Mendelian inheritance under sexual reproduction as a justification for the analysis. So path analyses could be used to give you causal inference because they had the causal anchors provided by genetics. Wright I think, would turn in his grave, and there's lots of his writings showing that he was already doing this, if he knew that these had been extracted, made into DAGs, and suddenly you can apply these just to any observational data, and as long as you can justify your DAG, everything's fine. That is not how this started. And Wright also, not only was he the progenitor of path analysis, which is with causal anchors, which is absolutely key, the second thing was Wright or his father, and probably actually a combination of both Sewell Wright and Philip Wright, his father, his economist father, introduced instrumental variables analysis. So Sewell Wright both introduced path analysis with causal anchors and instrumental variable analysis, which of course have become central to Mendelian randomization. Then a third component of the logic of Mendelian randomization comes from Thomas Hunt Morgan. Morgan also claimed to be the person who invented Mendel's laws. He said that he was the first person to put Mendel's first and second law together. But Muller, who was in Morgan's lab, said they all read Locke. So it's absolutely, they were told to read Locke's book when they got into the lab. So it's inconceivable Morgan didn't know that. But what Morgan introduced, crucially, was the notion of the instrumental gene. So this is completely distinct from instrumental variable. It gets confusing with the terminology. But what Morgan meant by the instrumental gene was that you didn't need to know anything about the physiology of the gene for making the inferences about genetic processes. As he was saying that it could be real or fictitious as long as the processes work in the way they do. You can actually use that for understanding physiology. So, these people were sort of doing Mendelian randomization to understand growth factors. Is there a universal growth factor in animals that sort of drives their size, or are there specific growth programs for lower limbs, upper limbs, et cetera, et cetera. They were using instrumental genetics to actually understand development, physiology, anatomy, etc. So that's the instrumental gene. And then the genius of R.A. Fisher, both one of the, if not the leading biostatisticians of the 20th century and also one of the leading geneticists of the 20th century. R.A. Fisher, in this brilliant paper, Statistical Methods in Genetics, the only time he talked about both statistics and genetics really formally in the same place, which was reprinted in the IJE. Everything that historical I talk about virtually has been reprinted in the IJE. R.A. Fisher said, genetics is indeed in a peculiarly favoured condition in that providence, with a capital P, has shielded the geneticist from many of the difficulties of reliably controlled comparison. The different genotypes possible from the same mating have been beautifully randomized by the meiotic process. Generally speaking, the geneticist, even if he, of course it's always he, then foolishly wanted to, could not introduce systematic errors into the comparison of genotypes because for most of the relevant time he has not recognized them. So Fisher recognized that this principle was what allowed genetics to be an experimental science that could talk about causality. So those of you here who come from a statistics background will say, oh yeah, but in statistics, of course, Fisher introduced the randomized controlled trial, which he did, and he introduced these in factorial studies in agriculture. as Fisher himself said, a connection between our two subjects which seem not to be altogether accidental, namely that the factorial method of experimentation, now of lively concern so far afield as the psychologists or the industrial chemists who are doing randomized trials, derives its structure and its name from the simultaneous inheritance of Mendelian factors. Geneticists certainly need not feel that the intellectual debt is all on one side. And Fisher's daughter, Joan Fisher-Box, and his last two PhD students are unequivocal that what Fisher then said in 1952 was not retrospective putting together, that the notion of randomized controlled trials came to Fisher from Mendelian randomization. So Mendelian, we always say analogy between RCTs and Mendelian randomization. Historically, Mendelian randomization came first, and so everyone should be talking about the RCTs. We've got this rather poor analogy of biology, but we've just got to see how this poor analogy works. So Mendelian randomization was primary. Okay, so then people actually trying to use molecular genetics to make inferences about modifiable risk factors, which is the absolute essence of Mendelian randomization. This is a paper that Shah Ibrahim and I didn't reference in 2003 because we hadn't read it. I hadn't come across it, but I came across later by Jerry Lauer. And he was using, it says N-acetyl transferase phenotype. That was because you genotyped by measuring the phenotype, because you didn't have genotyping technology then, but you knew that this was a Mendelian trait. And he was using this to make inference about modifiable exposures, in particular smoking and arylamine exposure in dyes, for example, and bladder cancer. These things do indeed, now we have to be absolutely certain, these things cause bladder cancer. But he used the fact that people who were less good at detoxifying these carcinogens were more at risk for a given level of exposure to say this strengthens our inference about that these factors do indeed cause bladder cancer. So this is an example of, in 1979, of using, so Lauer, they jumped straight to G by E form of Mendelian randomization. They bypassed the obvious, the blindingly obvious forms that we do. And with that, all of these, everything's reprinted in the IJE. Jerry Lauer became an environmental activist and still is, although he's pretty old now and stopped working in epidemiology. So he'd definitely be out with Extinction Rebellion. And I can't get it. There's no photo of him. He only puts up a photo of a monkey on his page. But as you say, he says, mutant genes do not cause neoplastic disease. And he goes on to say, the genes tell you about the environment. It's not that the genes aren't the cause. You've got to change the environment. So Lauer absolutely had the principle that I see as the key principle of Mendelian randomization. Then, so Shah and I didn't reference Lauer because we hadn't read it. We did reference Martin Catan because while the paper was under revision, I found Catan's letter in 1986 in the Lancet and then included that in the paper in the revision stages. And you'll know that we had Martin Catan here 2 years ago. He gave a fantastic talk on the origins of Mendelian randomization and then about nutrition and what genetics can tell us about nutrition. And this was a letter in the Lancet that had the brilliant insight of saying that in a situation where reverse causation seems key, where the early stages of cancer might lead to lowering cholesterol, then what at that time was a major controversy in population health and epidemiology, which was that observationally you consistently saw that people with low cholesterol developed higher rates of colon cancer and some other cancers. He realized that reverse causation could be avoided because there was an ApoE genotype which influenced cholesterol level and that the genotype would not be changed by the cancer. He didn't actually discuss confounding other than by early stages of disease, but the major issue in cholesterol and cancer, like the major issue in body mass index and mortality and many other things, is reverse causation rather than more conventional forms of confounding, even though reverse causation is confounding, but by very early stages, can be by very early stages of the disease process. He didn't have any data to look at this. He put this forward as an idea in in this letter. And of course, probably now we'd think we probably wouldn't choose ApoE as one's only instrument for cholesterol, LDL cholesterol, because it is a pleiotropic variant. That has also been reprinted in the IJ with a commentary by Martin. These are the citations on his letter. And it was cited once in 1988 and once in 1992, not to do with anything he said about reverse cause, but to do with, he put in a little discussion of ApoE biology in the letter, both of them just about the ApoE biology. And then by some remarkable bringing together of minds, how the world works through this, everything comes together. From 2003, it's been cited very highly, especially on the Atlantic, other side of the Atlantic, where this was the origins of Mendelian randomization that people had obviously known about, they just hadn't cited it before 2003. So then the term. So no one has actually used the term Mendelian randomization. I went through Fisher in detail, and he never actually puts the two words together as Mendelian randomization. So I came across the term Mendelian randomization in a paper by Richard Gray and Keith Wheatley in 1991. This is a brilliant paper, like Martin Catton's letters, without really any data, certainly not any data on strictly on the Mendelian randomization experiment they suggested. It was a review of how you could use observational data to understand whether bone marrow transplant reduced mortality in some forms of childhood lymphoblastic type leukemias, lymphomas, etc. Parents were not going to allow their child to be, absolutely understandably, to be randomized in a trial when you had these diseases, many of which had near universal mortality. You wanted to try any treatment you could try. So if anyone ever finds the term Mendelian randomization earlier than this paper, this is their quote when it's first used. I'd love to hear. They say, fortunately, however, the value of allogenic BMT can be assessed blindly without the need for formal trials by taking advantage of Mendelian randomization. So I heard about the term there, and then in a letter in 2000 writing about homocysteine and folate, I took the term from, I like the term, but of course it isn't, I like the notion of Mendelian randomization, but what they were doing was not the form of molecular genetics against exposure, against modifiable exposure that we now do. Their idea was more brilliant than that. Definitely more brilliant than anything that's been done, I think, since. What they said was, so for example, you have children with acute myeloblastic leukemia. Now, you can't randomize them to bone marrow transplant, but if they have a genetically compatible sibling, they're more likely to receive a transplant because the commonest source of a matched transplant is a sibling. And if they didn't have if they didn't have a genetically compatible sibling, they'd be less likely to receive a transplant. So if you analysed these by these groups, then that was like an intention to treat analysis. I think there was only one minor problem with what they suggested, and this was that they didn't take into account number of siblings. I suggested when we wrote a commentary on this 15, 20 years ago, that one would actually want to match by number of siblings because number of siblings is actually a risk factor relates to leukemia lymphoma risk, and also it could influence, the number of siblings could influence survival, so that would break the randomization. So this design should be used within sibling strata, and this design has been used now quite a lot of times, but it wasn't used at the time. So that's where the term came from. So this, as you see, this has nothing to do with the sort of molecular genetics being related to a modifiable exposure, really, with biomarkers that we think of. Yeah, it's cited, its citation also something exactly the same way from 2003. And this is a sort of, a representative citation. Apparently Graham Wheatley introduced a study design that used genetically determined differences in exposure to test whether a biomarker affects disease. And it's also used to say that Mendelian randomization had been going on since 1991. It hadn't really produced very much because it had been proposed in 1991, etc. So it's probably a good idea to read what you cite, I guess. So then in 2003, what did Shah and I say about Mendelian randomization? Well, firstly, we said that the fundamental principle of Mendelian randomization was gene-environment equivalence, because the purpose to us was that we were interested in modifiable exposures, things that you could do something about through intervening in the environment, but wanting to use genetics as a way of getting unconfounded estimates of causal effects of modifiable exposures. That was the idea, and the principle is, therefore, of gene-environment equivalence, that is that if a gene influences your exposure, it will have the same downstream effects as if you influence it in other ways. So in genetics, this is just what's known as either phenocopy or genocopy. These terms were introduced in the 30s and 40s, whereby you get two identical phenotypes. One is produced by a genotype, by genetic difference, and one is produced by an environmental difference. And a classic example of that is Hartnupp's disease and pellagra. So Hartnupp's disorder, identified in the late 1950s in a family who actually called Hartnupp, published in the Lancet. They used to use the names of the families to name these diseases. And this was a genetic disorder which resulted in poor absorption of amino acids, which resulted in a phenotype which was the 4Ds, diarrhea, delirium, dermatitis, etc. It just looked like pellagra. So the differential diagnosis for Hartnupp's disease was pellagra, and for pellagra occurring when people were niacin replete was Hartnupp's disease. But Hartnupp's disease is a Mendelian trait, and what produces it is poor absorption of amino acids, including tryptophan, which is converted to niacin. Now, when Goldberger, the epidemiologist in the 1910s, 1920s, did his heroic studies, he demonstrated that dietary deficiency clearly underlay pellagra, but he didn't live to actually see what the dietary element was. Armed with the evidence that an identical phenotype is produced by a genetic disorder of tryptophan absorption, would point to niacin as being important. So that's the genocopy phenocopy. So, Zuckerkandl and Villett, they said, no doubt all environmental effects can be mimicked by one or several mutations. I think that's going a bit far. I think being hit by a truck is probably not possible to mimic with genetic differences, but the point is that many environmentally modifiable exposures could be mimicked in this way. So we're all used to seeing the IV graph, the instrumental variable, genetic variant, and LDL receptor related to cholesterol. Relates to cholesterol, circulating cholesterol, you can relate that to the outcome, and that will not, those two associations, the IV and your exposure, and your IV and your outcome are not confounded, don't suffer from reverse causation. That allows some form of estimation of the causal effect. Or it allows you to strengthen inference about whether there is a causal effect of cholesterol on coronary heart disease. And you'll see this graph so many times today, so no one else needs to show this. This is the graph of the relevance assumption that your instrumental variable associates with your exposure, exchangeability, that there is no confounding, essentially by ancestry or factors relating to study entry, and the exclusion restriction, the variant doesn't have a direct effect on the outcome, which is not mediated through the exposure. Those are the assumptions. You've had them now, so no one needs to go through them. But those are secondary. The Mendelian randomization assumption, the primary assumption, is of gene-environment equivalence. That is, that if you modify cholesterol by, for example, dietary means, cholesterol-lowering medication, etc., if you change your cholesterol in those ways, that will have the same downstream effect on coronary heart disease as if it is influenced by genetics. Now, that is an assumption that needs discussing and defending in papers. It's more important in lots of ways than discussing the other assumptions need discussing. But this is the assumption if you want to make any inference to a modifiable exposure. In the cholesterol case, of course, it's rather strengthened by the fact that a genetic variant related to cholesterol absorption, Niemann-Pick C1-like-1, influences both cholesterol and coronary heart disease, and it relates to how the gut transports cholesterol. And it also happens to be a drug target, HMG-CoA, PCSK9, CETP, etc., etc. Many of the genetic variants, which are drug targets, influence cholesterol and relate to the coronary heart disease. And we know through randomized trials that drugs that influence those pathways influence coronary heart disease. So you have a basis for making that leap. But that is, if you want to use Mendelian randomization to say anything about modifiable exposures, that's the assumption that needs justifying. And Mendelian randomization, strictly linked to Mendelian principles. So in 2003, we said just straightforwardly that Mendelian randomization was, if you actually follow genotype down from parent to offspring, and then you follow phenotype and how that follows genotype, that is Mendelian randomization, and that then does have some analogy to a randomized controlled trial if you have parents and offspring and you follow down the genotypes. We said that's the design you want, but studies would be difficult to carry out because of problems in obtaining data from parents and offspring and because generally have lower statistical power. So the reason was that you just did not have any studies at all which would allow you to use this method except for things like folate in mothers and neural tube defects, for example, which is one of the examples we used. So that's in parent offspring. And we said MR in non-familial studies was a poor second best, and was in genetic association studies is approximate rather than absolute. But that in general could empirically demonstrate little confounding of genotypes by the conventional confounders, the behavioral, physiological, social confounders that confound observational studies. And the examples that we provided, and a very large number of examples over the years, many of these were not examples where you even could, in principle, do an instrumental variables analysis, such as, for example, does sheep dip lead to ill health in farmers? There's a 2019 review where these methods are discussed and have been applied now in larger studies, because because there was a genotype peroxidase variant which actually would detoxify what is in the organophosphates, and that if you have people, all of whom are exposed to sheep dip or all of whom are not exposed to sheep dip, then if the genotype relates to symptoms, and that in a way that suggests that those who detoxify less well have more symptoms, that would suggest that the exposure has an effect. You'll notice the logic of this is precisely the same as Jerry Lauer's logic. And we discussed many metabolic polymorphisms in cancers. Like Lauer's, I hadn't found Lauer's paper, but we discussed others. And the inference was from sort of rough effect estimates, where you could say, where you actually could you could make some effect estimate, through to just A increases the risk of B, but you have no idea of the magnitude of effect. Right the way through to A influences risk of B, but you don't even know the direction of effect, but you can say the genetics points to there actually being a causal effect, but you don't know its direction. The term instrumental variables wasn't used in the 2003 paper, and that meant that there are two IV estimates in it, but that meant that I rederived what I found out later was called the Wald estimator, and I misestimated the standard errors. So you can write into the IJE now and say, sir, I noticed in your 2003 paper that there are two instrumental variables analyses with misestimated standard errors, because I gave the data. So Steve's typing now. He's submitting his letter now to the IJE. You should say the paper should be retracted, obviously, because of this misestimation. So these are now sort of secondary features of MR that we discussed. In particular, I think it's important that Mendelian randomization is reverse genetics. So geneticists have bizarre terminology where when you go from disease, you take disease group and then you try to find out the genotype, that is called forward genetics. Even though maybe if you're an epidemiologist, that looks like reverse. And reverse genetics is when you take a genotype that you know does something and look out phenotypic effects. That is called reverse genetics. And indeed, when I first got excited about Mendelian randomization, I went and talked to Andrew Hattersley, who spent hours with me, hours and hours, putting me right on lots of things. And then at the end, he said, yeah, it's reverse genetics. And that was also where it came from. But reverse genetics was all, of course, done in animal models, et cetera, et cetera. There wasn't much human reverse genetics. So Mendelian randomization, if I had my time again, I would probably refer to it as human reverse genetics. Does that sound as good? Would you be here if this was a conference, the 4th International Conference on Human Reverse Genetics? Well, we'll see. Anyway, so, an example of Mendelian randomization, a very early example, that's work by Sarah Lewis, which sort of, illustrates already all the complexities. And of course, it was a two-sample MR. So all of the examples we included were two samples because we got the data from different sources, or because we knew the ALDH2 relates to alcohol, but not from these studies. And these were tiny case control studies of esophageal cancer in East Asia, where there's a genetic variant which leads to huge differences in alcohol consumption. And you show that the homozygous knockouts who really don't drink anything had a very considerably lower risk of esophageal cancer. But you found that the heterozygotes who had one knockout and drank an intermediate amount of alcohol had a vastly elevated, considerably elevated risk of esophageal cancer. And a naive MR analysis, you could do your IV analysis and you'd show that alcohol protects hugely against esophageal cancer if you use that comparison. But of course, ALDH2 null variant leads to accumulation of acetaldehyde, about 17-fold greater acetaldehyde in heterozygotes at a given level of alcohol than homozygous null variants, and acetaldehyde is a carcinogen. So there's no IV estimates in these papers. One of the IV estimates would make no sense at all unless you actually had acetaldehyde levels in the esophageal tissue, which we don't have. But these, I think, were like important studies on environmental causes of disease. So I've got 2 minutes for the future. 2 minutes on the future. So I think the future of Mendelian randomization is because of data opportunities and because of the depth of biological knowledge. So, some sort of ways of doing statistical analyses, these will help. But these are things, which the progress will come from these two things. So in particular, we have large family-based studies. So you can do between sibling MR. You can do you can do MR as it was originally introduced, which was from parent to offspring. You can do something which is analogous to an RCT. Between siblings is protected from both population stratification and dynastic effects. Dynastic effects are that you get your genes from your parents. Their genes, if they're things for behavioral or psychological traits, might be passed on by together with an environment created by those genes in your parents. So, if your parents have genes related to better education, there'll be more books in the household. If your parents have genes related to drinking more, there might be more violence in the household or whatever. So the genetic variants are confounded. So you can get around that confounding by doing comparisons between siblings. And you can do non-transmitted parental genotypes, revealing dynastic effects, and actually demonstrating that there are dynastic effects. To me, seems extraordinarily exciting because it tells you that there are parental influences on the outcomes without, again, doing an IV analysis or whatever. It just gives you that level of inference about the fact that there are parental effects on outcomes. You can do studies of disease progression. These all require individual level data. Do studies of disease progression, which will speak directly to treatments. Nearly everything we've done so far just talks to prevention, and prevention is vastly important, but we'd also like to use Mendelian randomization to look at treatments. For that, you need studies of people with disease, follow them up, see how MR tells you about how things influence progression of disease, which is very often very different from things which influence disease onset. Just think of lung cancer and smoking. You can do co-localization analysis, but there's the unprecedented depth of biological assessment, including tissue-specific data. And here I just advertise, first I'll show a slide showing that with ALDH2. So this is beautiful, I think. This is the first sort of example, if you like, of Mendelian randomization, where the outcome is actually looking at esophageal tissue and mutations in esophageal tissue, and you just do the same interaction of drinking with ALDH2, and you show what was shown right the way through down to esophageal cancer, but now you actually show it with mutation profile in tissue. And understanding how many of these other mutation profiles, and remember somatic mutations is a phenotype. I think there's about 60 or something somatic mutation profiles which have been identified. Most of those will presumably have some underlying environmental exposure. There's the alcohol one, the smoking one, et cetera, et cetera. Many of them are unknown. So using Mendelian randomization to understand what is the environmental modifiable cause of mutations which lead to cancer is incredibly exciting. And I'll just advertise a talk from Tom Richardson, whose slide I nicked on Thursday about tissue-specific Mendelian randomization, which allows you to say, when you do the MR, it's the phenotypic difference expressed in the tissue which is relevant to the disease. And this is Kurt Taylor's published paper on a sort variant and how this, the expression in blood does not show the same distinct pattern as the expression in liver, which is the appropriate tissue, obviously, for cholesterol genesis, and Lucy McGowan's work doing the same, with, again, published in the IJE, doing the same, no, Human Molecular Genetics, doing the same with respect to white cells and immune diseases. Now, the tissue-specific data, I think, is extraordinary because it really does increase inference. It really does increase plausibility of hypotheses when the processes that you're looking at appear in the tissue which the disease occurs in. Finally, we've all heard of the omnigenic model, introduced in 2018. This is T.H. Morgan in 1917. It might perhaps not be a very great exaggeration to say that every gene in the germplasm affects every part of the body, or in other words, that the whole germplasm is instrumental in producing each and every part of the body. So that's the omnigenic model in the 1917 version. The omnigenic model, which you'll know from Pritchard's work, is actually a multivariable Mendelian randomization model because it says the peripheral genes all have their effects through regulating core genes. The only way you can implement that, you can't implement that just with phenotypic data, because if you adjust for phenotypes, you introduce collider biases, which we'll hear about for 3 days, I'm sure. But if you actually set this up in a multivariable Mendelian randomization model, you can actually interrogate the omnigenic model through Mendelian randomization. So finally, what I see with Mendelian randomization is it can contribute to the triangulation of evidence. No form of evidence, not in a randomized controlled trial, gives you certainty. What inches you towards certainty is when a whole host of methods which have different potential biases which are orthogonal to each other, if you like, point to the same direction, and that really inches you towards making strong causal inference. Mendelian randomization in many situations will be available in two-sample data, for example, to look up whether your etiological hypothesis from your observational epidemiological study or your animal studies, etc., is borne out in data. So I increasingly hope to see Mendelian randomization, in particular the tissue-specific type data, of which we've inched up that much, in terms of what will come in the next 10, 15 years, will be just routinely used in triangulation of evidence. Just to advertise that the guidelines for strengthening reporting of MR studies have been, a very draft form of a summary has been posted, and you can put comments on that. Anyone wants to put comments. There's a special issue of the IJE, edited by Steve and Michael Holmes and Caroline, who are here, and Becky. And what more could you want than 2 whole pages of Mendelian randomization papers? So this will go live. Nearly everything is live already, but the whole issue will go live soon, and there'll be two more, because we've got so much, there'll be 2 more groups of MR papers in the coming year in the IJE.